Well, I'd like to welcome everybody to the inaugural Surinder Puri lecture. This is the first time we're ever doing this, and we're just overwhelmed by the family being here as well as all the people in the audience. I'm going to briefly say a few things, and then we'll have Dr. Puri come up and say a few things as well, and then Chris will get us started with the introduction of Dr. Alizadeh. We're very, very fortunate to have him here today to be the kickoff lecture for this series. So I met Mr. Puri a long time ago, and if you hear me calling him that, it's really not out of lack of familiarity, it's out of respect. I think I always called him that. He may have asked me to call him something else, but I always called him that. And he was a very kind man, a compassionate man, a quiet man, who even when he wasn't feeling well, never wanted to bother anybody. He was always about doing what he needed to do. In the midst of hyper-CVAD chemotherapy, I think he was still working pretty hard, and even during other therapy, going back and forth between here and Washington, D.C., because he loved what he did, and he really wanted to keep doing it. In the bio, you can see things like building bridges. That was really what he liked doing. And he was a light that influenced everybody around him. And it was a bright light, and it was a light that brought people together. He got to us, actually, through Dr. Majmudar, who many of you may know. We're very lucky to have him in the audience here today, an Emory pathologist who incidentally or coincidentally actually officiated my wedding before I even got to Emory. So that light from Mr. Puri spread and brought many people into his life as he went on. And when he passed to, as we say, a higher place and a higher being, and we were worried that that light may not be so immediately knowable, this idea and concept from Dr. Puri came forward. And it's so fitting that on his birthday, we're relighting that light on a day when that's what we do. We light candles to celebrate. And so we're very grateful to the family for doing this and to setting up this memorial lecture series in his honor. It's a huge opportunity for us. I know the last day has been just great for our team to be able to spend time with Dr. Alizadeh, who in my mind is one of the leaders in the world of lymphoma biology and classification. And that has been a great opportunity for us to continue to grow and be a better program than we've ever been before. So again, thank you so much to the family for allowing this to happen. With that, I'm going to stop and let Dr. Puri come up and say a few words as well.
Thank you all for joining us today for the inaugural Surinder K. Puri Memorial Lecture. Today would have been his 78th birthday, as Dr. Loniel mentioned, and I, along with my children, Shefali and Saurabh, felt that there was no better way to recognize his life and legacy than to officially launch this lecture series. Emory has been an integral part of our lives as a family. Our children grew down the street. From here, I did my residency and fellowship at Emory and worked at Grady. And Surinder and I received the finest cancer treatment possible here at Winship Cancer Institute. I'm a breast cancer survivor, and Dr. Lawson is here, I see. Indeed, when Surinder was diagnosed with stage 4 mantle cell lymphoma a little over 12 years ago, we were entirely unsure whether and how he would survive. But because we were so fortunate to be in the hands of world-class team of doctors and nurses here at Winship, Surinder not only survived, but for the next decade of his life, he really thrived and did everything in life he ever wanted to do. We as a family recognized that this would not be possible without the enormous advances in research and clinical care that Surinder was able to benefit from being treated at Cancer Institute here, and that too many other families, probably far less fortunate than us, watched their loved ones languish through the ravages of lymphoma and B-cell carcinomas. So it is our hope that Surinder K. Puri Memorial Lecture Series, by bringing to Winship the leading minds in research and clinical care to share novel ideas and foster potentially breathtaking breakthrough collaborations, will in some small way help accelerate the day when these diseases are just chronic diseases and not the terminal ones. Finally, before turning this floor over to Dr. Lonial, who has already spoken before me, I would like to thank Dr. Curran for supporting our mission, and also thank Dr. Alizadeh for taking time out of his busy schedule in being here with us as our inaugural lecture speaker. I do want to thank Dr. Lonial and Dr. Flowers for spearheading this today's lecture, and finally Jack, Ashley House, and Barbara for invaluable assistance in bringing this lecture series into being. And I will now turn it over, and Dr. Lonial, I do want to congratulate you again for the chairmanship of the department.
Thank you. And so I think we'll have Chris Flowers, who's director of our lymphoma program, come up and introduce Dr. Alizadeh. And for those of you who are wondering, the Indian food after this reception is not going to be a standard every Friday. Just, it's a special occasion. So.
Great. Well, thank you, Sagar, and thank you to the Puri family for setting forth this inaugural lecture. This really is a great occasion, and I can think of no one more appropriate than Ash Alizadeh to kick off this inaugural Puri lecture. Ash, like me, is a graduate of Stanford University. Go Cardinal! And I think we have at least one other Cardinal over here in the Puri family. Ash began his training at Stanford, graduating with both a PhD in biophysics and his MD from Stanford in 2003. He worked there with Pat Brown and spent some time at the NIH working with Lou Stout during that period, where he set forth really what was one of the preeminent papers in lymphoma, defining what is set the standard for how we define subsets of lymphomas using gene expression profiling, first defining diffuse large B-cell lymphoma into subsets that have both biological significance but also clinical significance. And that paper has really stood the test of time as one of the most celebrated, most preeminent papers in all of lymphoma. But he didn't stop there, fortunately. Ash has gone on from there to set new standards in lymphoma biology, both in defining subsets of lymphomas, both malignant and non-malignant B cells, and using those to help to define ways to treat lymphoma patients. And he'll talk to us today about one of those approaches, among many others that he's talked with us about over the days that he's been here. This is focusing on circulating tumor DNA and the role that circulating tumor DNA plays in cancer detection and its potential role in surveillance for cancer patients broadly and in lymphoma patients in specific. So Ash, without further delay, I welcome you to give this inaugural Surinder Puri lecture. Thank you.
Thank you, Chris. Dr. Puri, the Puri family, Dr. Lonial, Dr. Flowers, and the audience, thank you for the kind invitation, the honor to be with you and tell you a story that I hope will teach us about a tool that perhaps can be useful to us. We'll be talking about personalized cancer detection and monitoring using something that I didn't know about 10 years ago, but learned a lot about in the last few years, and I'll tell you a little bit about what we've learned about this interesting way of measuring disease in the last few years by taking two examples of the commonest blood and solid tumors as applications of the method. So I'll start, the outline of my talk is to give you a little bit of background on the molecule, circulating tumor DNA, describe to you a new method that we've worked on developing called cancer personalized profiling by deep sequencing, or CAP-seq. Initial applications of this method to non-small cell lung cancer, the commonest solid tumor, initial applications of it to the commonest blood tumor, or large cell lymphoma, some ideas for how this might extend to other tumor types in highly personalized ways, and give you a vision and perhaps future directions for translating this biomarker. So historically, when we think about personalized precision cancer therapy, and I'm not talking about the design of drugs, but I'm talking about an idea for taking apart heterogeneity among cancers. We're used to knowing the difference between a breast cancer and a lung cancer based on different parts of the body they might grow in, but we are increasingly refined at understanding the things beyond just the locations of these cells that are captured in the molecules that contribute to that heterogeneity, DNA, RNA, and proteins. And we have found ways of measuring that through the genome, through the microenvironment of the tumor, but now increasingly in ways of doing that non-invasively through the blood. And the idea here is that through these refined ways of measurement that we can use our tools in a sharper way. And that is what we call precision personalized therapy of cancer, and I'll tell you a little bit about that today. So we'll be focusing on circulating tumor DNA. So all cells in our body, as they turn over and die, release DNA that needs to be cleared. And that DNA is largely enters the circulation and is digested by DNases made in the pancreas and other parts of the body. And this DNA is admixed from whether tumor cells or healthy tissue with the cells in circulation in small amounts. Now this is something, the free DNA in the blood is something that was discovered by two French scientists in the 1940s, but we don't talk about it a whole lot until recently. And it wasn't until the 1970s that it was recognized that the levels of this DNA is higher in patients with cancer. But the reasons for that being higher were not clearly established until the late 1980s, when some mutations were found in this DNA that circulates in the blood. But the levels were so astonishingly low that we've had to wait to translate this technology. So what is this DNA? The DNA, the major species, is about 175 DNA bases, A, G, C's, and T's, or one nucleosome, or technically a chromatosome wrap. So the DNA is wrapped around some core proteins, the histone nucleosome complex. This is my blood and cfDNA distribution of DNA that's in my blood, roughly around 170, a disome, a trisome, roughly 5 nanograms per milliliter of this material circulates in our blood plasma. Higher levels, as I mentioned, can be seen in patients with cancer, but in various non-malignant conditions, including infection, inflammation, trauma, etc. And through transplantation studies, where donors and recipients are mismatched by gender, we can say where this DNA comes from. And if you do that from hematopoietic stem cell transplantation, you can see gender discordant DNA. And if you do solid organ transplantation, you can do the same. And we think something like 80 to 90% of this DNA derives from the hematopoietic system, leukocytes as they turn over. Now this DNA has an extremely short half-life, less than 2 hours. It depends on the exact species you measure, but for the major species as measured by CAPS-seq, we think our half-life is about 40 to 45 minutes. Now, we measure a lot of things in the blood. This is courtesy of Wikipedia, and there are a lot of hematologists in the room. So there are things that we measure that are at the very high range, like hemoglobin and albumin, where we're measuring things in grams per liter. There are the hormones that are much lower partial concentrations, like thyroid hormone, that are in picograms per milliliter. There are the protein biomarkers of cancers, like prostate serum antigen, CEA for colon cancer. These are somewhere in this lower range. And circulating tumor DNA spans this lower part of the concentration range. But unlike these molecules that we measure using analytic methods that detect them directly, we have something unique in circulating DNA, in that we have the power of the polymerase chain reaction in amplifying this signal and being able to detect it at very low partial concentrations. So as I mentioned, this is a very broad range from less than a fraction of a cell. You know, a cell has 6 picograms, so less than a fraction of a cell per milliliter to greater than 10 nanograms per milliliter. And the concentrations are usually less than 100 picograms per milliliters, or 10 to 15 cells per milliliter of blood. And in terms of other things that we measure, only hormones fall into this range. So what are some problems for detecting this DNA? Well, as I mentioned, concentrations are low. So for a defined amount of blood, we are limited by the number of molecules that we have available to us. We need an ultra-low detection limit that allows us to find a needle in a haystack by not being fooled by the errors that the enzymes make in amplifying DNA. And we need a method for being able to apply this broadly to patients, because cancer is diverse and mutations are not the same in every cancer. So these are the problems that we deal with. To tackle this problem, we set out to look at all the data that's been generated through the various public and private genome and exome sequencing efforts. And the approach we took is to say, let's take a population of patients with a given cancer, and let us find the business end of that cancer through the identifying recurrent mutations that are shared across patients. And this is not simply taking the genes that we like based on their recurrent frequency alone. It's using an algorithmic framework that tries to say, what's the smallest space in which I can capture the most mutations so that I can profile a given patient. And this selector, or this diamond, this red selector, is then used to define the mutations of a person's tumor, and using that signature of mutations, recover them in the blood plasma and monitor them in cell-free DNA during a diagnosis and during treatment of the patient. Our goals were to apply this to a broad population of patients, and we started with lung cancer because at the time the dogma was, this is not one disease, it's 100 diseases, and it's hard to imagine that we could use one genetic test to monitor it. And we wanted to do this in a way that allowed us to measure DNA at very low limits, and that we could capture the diverse types of somatic alterations that you find in DNA. Single nucleotide alterations, insertions and deletions, copy number alterations, and gene fusions, translocations. All the variety of bad things that can happen to DNA that can drive it. And we designed this initially in the original description for about 139 genes, about 500 some odd chunks of the human genome, for 125 kilobases. This is a relatively small fraction of the genome, much, much less than 1%. And we said we wanted that to be done economically. Now we take DNA, whether it's from the tumor or the cell-free fraction of the blood that I showed you before. We take these fragments and we make these things called libraries. We then go fishing out the pieces that we're interested in using pools of biotinylated oligonucleotides to pull out the business end of each cancer genome that we capture, we sequence, we find the alterations as a needle in a haystack, and we keep track of that. And these are the nuts and bolts of how we do it, but we sequence at very, very high depth, in some patients at 100,000x, to leverage the sensitivity that you can achieve. Now this is a graphic from an important paper from Bert Vogelstein and colleagues in the journal Science about 3 years ago, summarizing what we knew about cancer somatic landscapes at the time. And this really hasn't changed a whole lot since then, so please allow me to use it as a framework for discussion. Here we're looking at the common adult human cancers and pediatric cancers in terms of the distribution of mutations, just in terms of the frequency of mutations. And so at the top of the list are colon cancers that are microsatellite unstable, that can have over 1,000 mutations per cancer genome that change the protein coding sequence of a protein. Lung cancers fall up here, and we'll be focusing on those lung cancers from patients who are known to have been smokers. They're never smokers with lung cancer that have fewer mutations. And I'll also talk about lymphoma that falls into the intermediate range. So we're going to be talking about this broad range of human genetic alterations in cancers. So we started with non-small cell lung cancer. This is the commonest cancer and leading cause of cancer-related death across the world. Over 200,000 Americans diagnosed with this disease. And we said, how well does this work? And we started with a very small collection of patients, and this has been published now, but we've now applied this to hundreds of patients, and the results hold. And what we were strikingly surprised by was how sensitive and specific you could apply this method to detecting tumor in patients, whether they were early or advanced. So we For all stages, we had about 85% sensitivity for detection and 96% specificity for unique detection of the disease across very low amounts of DNA in blood samples, 40 blood samples from these patients, and across a range of concentrations of that DNA. Now, what we weren't expecting is that this DNA would correlate with things that we could measure, like how big the tumors were. And this is the tumor volume of these patients compared with the concentration. And you can see this is largely a linear correlation, and this is held across other tumor types, including lymphomas, perhaps with the exception of highly localized tumors that contribute less to DNA in circulation. Now, the cancer experience is a timeline, and the disease burden varies as a function of the stage at which a person is diagnosed, the treatments they receive, and the way with which the tumor responds. So if we were to imagine a hypothetical timeline over which circulating disease burden goes up and down, one can imagine that if a patient at some point has targeted therapy for a given marker and resistance emerges during targeted therapy, one can monitor for it during that targeted therapy period. And this we have done, and we have a number of studies that I won't talk about, where we've been able to identify mechanisms of therapeutic resistance during treatment with targeted therapies. The second is disease surveillance in the context of disease that is not measurable radiographically when we are looking for relapse in a sensitive way. And the ultimate holy grail is to allow us to detect diseases, particularly solid tumors, early during a screening interval that might help us prevent advanced forms of cancer through maneuvers that can eradicate tumors. But one of our major goals in that final exercise and in the exercise of monitoring is to be able to do this without biopsy tissue. So we have a given patient that we take, and we have a blood draw from which we would like to genotype and monitor in the absence of tissue. And this is really a difficult challenge, and we've done a fair amount of work, and it looks pretty promising, and I'll tell you a little bit about that experience. So the main factors that we've challenged with are the molecules recovered, the mutations covered, and the technical artifacts that keep us from being able to go very low. And this is captured as follows. This is a complicated graph, but I'm going to walk through it. The principle of this method is that these DNA molecules are in pieces, and they circulate in the blood in such a way that if you have a blood tube of 10 milliliters, with 5 milliliters being plasma, and 30 nanograms of cell-free DNA is present there, and let's say that there are 5,000 genome equivalents of which we recover half. If one had a single mutation that they were following, if the fraction was 0.1%, you could be very confident that you would detect that. But if the fraction were going much lower, the likelihood that one would detect that, this is simple probability rules, drops. As you add mutations, you improve the odds of this, and this is what's motivated. The statistical observation is what's motivated the designs of our selectors towards increasing the number of mutations to monitor patients. And this principle also is the same as having more genomes to play with. So this is, if you look at the same y-axis and consider the number of tumor reporters versus the likelihood of detection, as you have, so 5,000 is 10 milliliters of blood, 20 milliliters of blood, 40 milliliters of blood, and lower what the likelihood of detection becomes. So as mutations are higher and genomes are higher, you can go lower in seeing this. Now, so this is a probabilistic framework, and there's an equation that can be derived that explains the rules for this framework. And if you go from 1 mutation to 8 mutations, you can, that's our working unit for lung cancer, you can have an increased probability of detecting that. Now, these are the theoretical limits of detection, but there is this gray mountain that we've had to fight. And this gray mountain is the errors that we observe during sequencing of DNA. The lower you go, the more errors you observe. And this is what we bumped into in the original description of the method, which was as you were trying to go lower and lower, instead of being on this line of expectation, we weren't able to go lower and lower because we were bumping into these errors. This is the blood of 30 healthy adults, including myself, sequenced at various steps. You can find errors in all of us, but this is what we need to get rid of. Now, we've made a lot of progress towards this. This is a highly technical concept that I won't belabor, but we've combined two approaches that we call integrated digital error suppression, which uses DNA barcodes to keep track of molecules, and a statistical method for polishing that allows us to go from this red line to this green line. And that green line allows us to now go to unprecedented limits of detection. These are for solid tumors where we really need to be, and for minimal residual detection where we need to be if we're going to detect fewer and fewer cells in the body. Now, I mentioned this idea of doing it without a tissue biopsy. We do usually, if you have a tumor biopsy, you can track the disease, but what about when you don't have a biopsy? Can we draw a blood draw and recover mutations and be confident that these are bona fide mutations from the tumor? So we've applied this to lung cancer for non-invasive tumor genotyping. So a substantial fraction of patients with lung cancer today do not get tested for mutations, depending on which part of the world they're in, for targeted therapies that can significantly extend their lifespans. In China, it's expected that something like 20% of patients are tested. In the US, the statistics are less well known, but we think we test about 60 to 70% of patients with lung cancer to get targeted therapies. That's not true at Emory at Winship. It's not true at Stanford, but lung cancer patients don't only visit those two institutions. So we applied this to a group of patients with 100 blood samples of patients known to have mutations in EGFR. And we said, could we detect them in their blood? And we used sensitivity and specificity analysis. And the sensitivity and specificity for these patients, admittedly advanced patients, for detecting the DNA in their blood is 100%. Now, what if we didn't know about this in the first place, and we wanted to genotype the mutation? So we've done that across a range of mutation types, including the two dominant mutations that drive EGFR to be activated, the resistance mutation called T790M that mediates resistance, but also is a target of next generation drugs. And we said, let's pretend we don't know anything about these mutations in the EGFR kinase domain and see if we can detect them. And we applied this to a number of patients. You can see, depending on the subtype of mutation, what they are. And we were able to achieve reasonably high variant level and patient level sensitivity and specificity using this approach, which we call integrated digital error suppression. So high 90s, where I think this is where we need to be if we're going to use these tools in patients. Now, what about tracking the disease? What I'm going to show you is a number of patients where the x-axis is time, the y-axis is the DNA in the blood, and the red are the mutations that were discovered in the blood and monitored in the blood, and the blue is the mutations that were detected in the tumor. And each one of these 8 patients with lung cancer is being monitored now in 2 different ways, without knowledge of their tumor or with knowledge of their tumor. And you can see, by and large, you get the same answer, whether the disease is responding to therapy and things plummeting and staying plummeting, responding, failing, and going away, without any imaging here. We're simply just monitoring the blood. And I think this shows you that you can do this without access to tissue, especially in patients in whom access to that tissue is difficult. So we talked about lung cancer, right, as a difficult disease, which is heterogeneous, and we've turned it into a single analyte that we can monitor. So that's a solid tumor. What about a liquid tumor, or a liquid tumor that likes to behave sometimes like a solid tumor, non-Hodgkin's lymphoma? And this is a disease, as Chris mentioned, that I've studied for a long time. For this, we focused initially on diffuse large B-cell lymphoma. This is the commonest blood cancer that is curable in a substantial fraction of patients, but patients who experience relapse or have refractory disease have a very poor prognosis. It hasn't been clear that PET-adapted therapies have been truly effective for these patients. We heard a little bit about an interesting strategy from a group in Australia that may have changed that a little bit, but in general, this has been relatively disappointing to use imaging. So we've been particularly interested in using ctDNA in these patients in a broadly applicable tumor-specific way to measure residual disease, detect relapse, and measure tumor burden and heterogeneity, and watch the DNA as a way to look into the tumor. We began this by looking at a single gene. Lymphomas are special in that they have rearrangements of their antibody gene, the immunoglobulin gene, and this can be a barcode that you can monitor. And we worked very closely with a group that developed this technology to apply to a group of patients with lymphoma. And we focused on this single gene, and we noticed that it had high specificity, 100% specificity, and reasonably high rate of tumor detection in patients who had accessible tissue. But on the average tumor that was fixed, it was troublesome, only applied to about 80% of the patients, and had somewhat limited sensitivity in that patients who ultimately relapsed were often undetected for many, many time points during their remission period, and could at most follow one or two things per tumor. So to overcome that, we applied the same method that I described to you before, where we took a population of about 1,000 patients with lymphoma from the public domain and from patients that we had sequenced, and looked for things that were common between them in the DNA alterations, single nucleotides, insertions, deletions, the genes that translocate, BCL2, BCL6, and MYC, the antibody genes, and the structure that changes them, in targeting about 250 or so genes, 1,000 chunks of the genome. And we did this in the same way by monitoring a given patient at diagnosis, during their treatment, and before preceding their progression or relapse by monitoring the DNA longitudinally. So what about the idea of simply trying to genotype patients at diagnosis and relapse when we know them to have disease? So we applied this to a group of 76 patients, in whom 45 of whom we had match time points in blood, and we were able to detect mutations in all but one patient in the tumor, and the vast majority in the blood, with on average 70 mutations in the blood and 140 mutations in the tumors. Something we weren't expecting was that we would be able to capture the translocations of these patients in both compartments using the same approach. And even though we weren't trying to do this in a very, very precise way, we had reasonably good performance of roughly 80% genotyping, and I think these could be even improved if we did the assays better for these genes that we use clinically to change our therapies, detecting them both in the blood and in tissue, and we're looking at the concordance between a method that we use in standard fashion called FISH, and this blood or tissue detection using CAPC. And this was the success rates. Now, Chris and Sagar mentioned this idea of tumor classification. In large cell lymphoma, one of the things that we defined some years ago are two subtypes of this disease. And the two subtypes of this disease behave differently clinically, but we've had a difficulty in measuring this in a clinical context. So we set out to say, can we measure this using DNA? And we were able to do that in these patients from their tumors or their blood samples. with the highly significant association with the patient's survival and highly concordant measurement, whether you compared it in the tumor or the blood plasma. This is not something we were expecting, and we're going to be even refining this further. Now, what are the various parts helping us do? So I told you about the antibody genes, and the antibody genes help us detect about 50% of patients, but in a highly specific way. What about the translocations? They improve things by about 30% for sensitivity, and the specificity remains the same. But as soon as we look at the somatic alterations as single point mutations, that's where the sensitivity boost really comes in, such that we can detect every patient in a sensitive and specific fashion. Now, tumors are villages. They are villages of clones, clones with machine guns, right? These are a competing a group of cells that are at various levels of fitness, and the somatic mutations in them can give us a picture into how heterogeneous they are. We saw something that we weren't expecting, in that if we looked at these clones, by each one is a mutation here in the tumor versus the plasma. Here's a patient in whom the blood roughly has the same amount of DNA as the tumor, highly concordant. So you're basically able to look into the blood and infer the genotype and the clonal architecture of the tumor with high precision. Here's a patient who has much less DNA in the blood, 1 or 2%. And again, the concordance is like this. So these are 2 patients, and this is the result summarized across 40 patients, where we're seeing the vast majority of patients have correlation between the 2 compartments. The second thing we observed is that the DNA is, again, a biomarker for disease burden. So for lymphomas, we often measure a protein called lactate dehydrogenase, and in many patients it's normal, but in nearly all of these patients we can still detect when they're below normal circulating tumor DNA, and that is highly correlated when the LDH is high. We also use STAGE, or the International Prognostic Index, to have patients with low, intermediate, or high risk, and the DNA is stepwise related to that. Here are the other strata. And that concentration is just a simple measurement of circulating tumor DNA is associated with the survival of patients. So we talked about this biomarker at diagnosis of disease. Well, what about during therapy? So let's focus on that. We're talking about the early interval during treatment of large cell lymphoma. So to do this, we looked at a group of 30 patients, in 12 of whom we did a very close observation of their DNA dynamics early during therapy. These are patients that had a variety of risks, a number of prior therapies, and a number of treatment regimens. We separately had a validation cohort, and our total patient population was 33 patients. And the first observation was this graph that we call a spider plot. On this graph, each patient is a line, and each dot is a blood time point. And in color, we have captured what ultimately happened to these patients in terms of their responses to the therapies that they were given with curative intent. And the green represents those patients who achieved remissions. In red, those who did not and had their disease progress. And in yellow and orange are second and third base kind of responses that are partial or stable disease. And you can see within days of start of therapy, the DNA plummets, and those drops are quickly able to separate the patients by whether they were green, orange, or red, such that by the first day of their second cycle of therapy, you can essentially tell who's who. So we set a threshold of 100-fold or a 2-log reduction from this patient population of drop in DNA, and we then extended it to the whole cohort. So the y-axis here is how much DNA changed after one cycle of therapy, and again, are those outcomes. Green is what you would hope to achieve, yellow is not quite, red is not what you want to achieve, and this within a month of starting treatment, you can have a high precision for measuring what ultimately happens to the patient for overall response and complete response. Now this was, we weren't expecting this to work within this population as well as it did, but we could predict the progression-free survival and overall survival of these patients in a training validation framework that was independent of the other things we know are clinically important in these patients. How much therapy they'd had, how advanced their disease was, what cell of origin type they had. But this is still not perfect. 2 out of these patients relapsed after a complete response. And all these patients with astraces, a green patient here, and these yellow patients are somewhat misclassified. So the classification is not perfect, and this is something we're trying to refine using a variety of ways that we're working with Dr. Flowers on exploring and modeling frameworks that are going to require much more thought. So we've talked about the early treatment responses. What about progression and relapse after a patient has achieved the radiographic standard of remission? So to do that, we've looked at a number of patients, all of whom relapsed. Each line is a patient. Each gray area is when they were treated. Each circle is a blood sample, and red and white reflect whether circulating tumor DNA was detected. In this red line is the time at which they relapsed. They all relapsed, and we are now going to look back at when we could detect them. So in 12 out of 12 of these patients, we were able to detect relapse when the PET scan told us that the patients had relapsed, but that's not much of a feat. For 7 out of 10 patients, we were able to detect their relapse before the radiographic time point, depending on when blood was available. And this was with roughly 5 months of lead time in these patients. In fact, this was specific in that we had 13 patients, not shown here, that were in complete remissions for more than 5 years, and none of whom we could detect residual circulating tumor DNA. And this was associated highly with survival. And I'll take you through one of these patients myself. This is a young woman I took care of who we lost, sadly. This is a 42-year-old woman with a stage 2 large cell lymphoma that by genotype was supposed to do very well. IPI of 1, germinal center subtype, but this large mass in her abdomen. So 51 mutations were identified in this tumor and in her blood plasma during therapy and relapse. So here we're going to look at this on the x-axis in tumor allele fraction and the y-axis in her blood. So when she was diagnosed, you can see a high relationship between the two, and we can find several subclones by the distribution of alleles. Now let's see if I can get this to. It's a video, but I don't know if I'm going to be able to make it go. So we're going to watch her during her early course of therapy. This is a chemotherapy with R-CHOP, where she achieves a partial response, but nonetheless has disease detectable. The disease progresses, and we can see all the clones reappear. She gets salvage chemotherapy, achieving a complete response, but nonetheless still has detectable disease, even though she's thought to have a complete response. And the disease progresses and vacillates, where new clones emerge, and we see progressive disease that she ultimately succumbs to. Now, these clones you can watch in another way, and you can say, can I use these clones to inform therapy? Some of the mutations that arose could be thought of as novel therapeutic targets if the right trial were available to assign this patient, but that is the subject of another talk. Here is another patient of mine. This is a patient with follicular lymphoma who was diagnosed in December of 2013, in whom we saw differences between the tumor and the blood. We found things in the blood that weren't in the tumor. And the blood was so different from the tumor that we had to wonder what was going on. This patient received rituximab and achieved a partial response, but with a mass in the abdomen remaining and growing by 9 months later, which was then repeat biopsied. That biopsy showed transformation of low-grade to high-grade lymphoma, and the blood mirrored that transformation. The colors here represent the mutations and the allele fractions. And the blood at relapse looks very much like the blood at diagnosis. That tells us about something occult that we could not see in looking at the tumor. Here's another patient who was part of a clinical trial that led to the approval of this drug, idelalisib. And this patient, again, has experienced transformation, and you can see here is the two tumors before and after, where certain mutations emerge and certain ones drop, and you can watch the same thing happen in the blood. So we've compared a group of these patients by both immunoglobulin sequencing and this CAPP-seq, and found CAPP-seq to have advantages both for tumor genotyping, detection of pretreatment DNA in this blue box, how many more patients we detect, immunoglobulin measurements, and prediction of relapse. So we are very excited by this and hoping to now apply it to more patients. So I hope to have convinced you that, that one can robustly genotype tumors, find the mutations non-invasively at the pretreatment time points, and for molecular residual disease during surveillance. We think there are advantages to this approach of liquid biopsies in patients who achieve radiographic remissions, and we think that this could allow us to predict relapse. And we think that we could also measure subclones and dynamics during therapy that might tell us about response and resistance and the development of new therapeutic targets. Okay, so this is particularly easy for tumors that have many, many mutations. But what if the tumor does not, or it's very difficult to infer what the mutations may be? So I described to you the off-the-shelf approach, where we used a group of patients to find something that would be applied. But another way to do this is to do it in a patient-specific manner, where you genotype the tumor using whole exome sequencing or whole genome sequencing to discover the mutations and make a custom tool to monitor a patient, and we've done that. Now, we did this to see how low you could really go, and really you can go to 2.5 in a million molecules using this approach. This is using a lot of mutations from a patient with glioblastoma in a spiking experiment, where if you leverage many, many mutations to monitor with higher and higher precision. We applied this to a patient with a brain tumor, glioblastoma. This is not a tumor that circulates in the blood at significant levels as a cellular tumor. We don't detect metastasis. This young boy had a glioblastoma treated with surgery, radiation, chemotherapy, and ultimately a vaccine, EGFR vaccine. Sadly, he had disease relapse 485 days later. We had sequenced the tumor at diagnosis, and we separately sequenced the tumor at relapse. And we found some alterations that were present in his tumor at diagnosis and relapse exclusively, but a lot that were unique to the relapse tumor. And these mutations were tracked in the blood and his cerebrospinal fluid during his treatment. And what you see here is each row is one mutation. Here's his tumor at diagnosis. Here's his tumor at relapse. Here's his cerebrospinal fluid, some healthy subjects. And what you notice here is the mutations that are shared in both the diagnosis and relapse tumor, the mutations that evolve at relapse, and the fact that we can detect them in the blood of this patient some 200 and some odd days before radiographic relapse. So this really shows you how precise you can be in detecting disease in even microscopic non-circulating fractions. We've done this with other tumors. Here's another tumor with a woman with ovarian cancer, stage 3 ovarian cancer. My mother's a survivor of this disease. This is a patient in whom we sequenced the tumor and the germline, identifying that she had a BRCA mutation. that was not previously known, and we found 45 mutations that were tracked during her course. And here's her course from her original surgery and debulking and various types of chemotherapy, and this is the biomarker that is typically followed, one of those protein biomarkers that I showed you early on, CA125, that goes low. And you can see here, we started monitoring her blood here at this time point, and we've been able to track her disease very nicely during the course of therapy. So what do we see as an application of this method? What is the vision? What we're hoping to do is that we make measurements all the time, and oncologists use information to have informed discussion with their patients. But we hope that that can be made easier through access to a non-invasive way of measuring the disease, detecting circulating tumor DNA, measuring the disease precisely, and generating the clinical evidence base to make treatment recommendations that will ultimately improve patient outcomes. Now, this is one idea for how that might be done. This is looking at Medicare spending on imaging. And this looks at the same costs at Stanford University. Here, the y-axis is the mean cost per Medicare beneficiary from 1999 to 2005 for imaging for Hodgkin's, non-Hodgkin's lymphomas, lung cancer, colorectal cancer, breast cancer, et cetera. And you know, they say 4,000 here, but at Stanford it's about 10 times that is what patients are charged. And the utility of these approaches is not clear. It depends on the exact disease context. But one can imagine that if you can do this better, you might help the population in a sharper, more refined way, and perhaps less expensive way of specifically and sensitively monitoring the disease. This would apply to many cancers if the tool was the right tool. And we also see a vision for looking at patients with locally advanced disease, following them during the course of therapy, and being able to tell them early on whether they're going to do well or not so well. In patients with advanced disease who are getting targeted therapies, to watch clones change and tell them, are they likely to respond or fail with the drugs that we are administering. And in the relapse setting, for the likelihood of recurrence, and to study new therapies and measure things very early on during the course of therapy, within days to weeks of treatment, given the short half-life of this molecule and the way in which it relates to tumor burden. So what are some predictions that we might go out on a limb for? I'm pretty confident that the technical improvements will allow us to improve the sensitivity and specificity of these tests. We still have a little ways to go, but I think it's not too big a leap to say that this will happen. I think these costs associated with sequencing the DNA will also come down in parallel with the cost of sequencing. And I think it will become routinely available as a clinical and personalized biomarker for common cancers, for the genotyping of tumors, early response monitoring, residual disease detection and surveillance. The medium-term projections are that I think that in early detection of some cancers, this will likely be possible, perhaps in the context of secondary screening to reduce biopsies, and maybe someday primary screening of cancers. I don't know how long it will be for this, but we already have a framework for this using DNA detection in the stool for colon cancer, so the idea of doing the same in the blood seems like it could work. This will require a lot more work, including technical improvements, the careful selection of tumor types and the patients who are at the right risk population, large clinical studies, and that is work that we have to do together. So with that, I'd like to take this time to acknowledge the people who made this happen. We've worked on this for some time, and it's been a collaboration largely between my lab and Max Dean's lab, my partner, and some people who have stuck with the project from the very beginning, in particular Aaron Newman, who's been involved from the very beginning, about 4 years ago, Florian Scherer, David Kurtz, Alex Lovejoy, Dan Klass, other members of the laboratory, our funding sources, most importantly, our patients and their families. Thank you very much.
Very nice talk. Congratulations on your work. Two questions. Number one, tumors are not homogeneous. Tumors are heterogeneous. And within one single tumor, we have different clones. And one of the questions I have is that you have a panel of mutations to detect this disease. But how does that represent for the whole population? And your clear example was the patient with follicular lymphoma that had a richer transformation from the early on. And the second question is, how about the effect of chemotherapy on your panel? Does that chemotherapy change the profile of mutations that you see?
Great. I'll repeat the question. Dr. Bernal was asking whether how heterogeneity is captured. So what we were surprised by in those allele fractions that I showed you is that these village of tumor clones are mirrored in the blood, suggesting there's a quorum that mirrors their allele frequencies, but that we can only measure what we detected in the first place. So if we didn't include the right mutations, we would miss them. So to overcome that, I think the approach is to take a population of patients who have failures and include them in the original design. The other possibility is to say, let's open the lens, and instead of looking at a smart part of the genome, at a much bigger part of the genome in the candidate space, and we've done that. So I hope that I answered your question.
So is there enough DNA in the blood to do the whole exome and get
Yes.
pretty good depth of coverage?
Yes, so if it's a function of cost and allele fraction. And so there's a group from the UK that has done the work. And when you have tens of percent, you can. But very few patients are at those burdens. In advanced metastatic patients, there are. So in fact, for many patients in that context, you will have a better representation of the tumor in the blood than in the tumor.
Okay. Yes. Other questions? We'll do this and then
How sensitive is CAP sequencing being detected early in the process or recurrence as opposed to current PET scan?
Yes, so the 12 patients that I told you, all of those patients were in radiographic remission when we were monitoring their blood. So every patient who relapses with a PET scan is positive, so 100% sensitivity at time of relapse, okay? And that's not a whole lot different than what we presented for immunoglobulin sequencing alone. Now that's at the time of relapse, when you know the patient has relapsed by a PET scan, and we tend to do PET scans a lot at Stanford, so it's a pretty dense interval. But the lead time is what you're really interested in to perhaps intervene in the context of lower burden. And there we think we have a few months lead time, and with more sensitive methods, perhaps longer.
This is slightly off topic, but I was wondering if this kind of technology could be used to measure T-cell receptor repertoire in sort of the setting of GVHD to look for the responsible clone, or to start getting a little bit more information about
Yes.
really serious immune function.
Immune function. That's a great question. I've been trying to convince a couple fellows in my lab to work on that very same problem, but they're not interested. We're chimeras when we get transplants, and our T cells see foreign antigens. So could you adapt CAP-seq to look at those antigens and the T cells? For GVHD, for instance, if we have difficulty knowing how long to keep a patient on immunosuppression, but if you could watch the tissues that are being attacked of the host and of the graft in real time using polymorphisms that distinguish them, you could do the same thing. And in fact, similar technologies are being adapted to solid organ transplantation with very, very high, but it could definitely work.
Okay.
You compare the tissue mutation with the blood tissue. In the early stage, they match well, and later stage, they probably didn't match well. I think that maybe metastatic setting. Have you compared the metastatic tissue with the blood tissue?
Yes. So the question is, what's the correlation of the mutations that you detect in the blood versus primary versus metastatic tissue. So we've done a little bit of that. There's a fairly extensive literature in solid tumors about the concordance of genotypes between primary and metastatic sites for actionable mutations. Epidermal growth factor receptor, KRAS, etc. And there, the match, you'd be surprised, is not as high as you would hope. It's not 100%. It's somewhere between 80 and 90% for most of those lesions. We see similar correlations when we compare the blood plasma to the tumor, the primary tumor, and at relapse. But we find many things that end up being the likelihood of detecting a mutation non-invasively in the blood. It has a much higher positive predictive value of it being in the tumor than vice versa. So the tumor is a geographic confine that tells you about the local clonal dynamics. But in the blood, you capture the body experience. So if you detect something, it represents a clone. You just have to do it with specificity.
You may get an amalgam of all the different
Yes.
genetic sites.
You will not know where it comes from. And that's the challenge then of if you're doing focal therapies, how to use the information.
Okay. All right. Well, thank you very much, and we. There is food out there, so 1 second. Do you want the kids to come up with you as well? Whoever you want.
That's enough. That's enough.
Hi guys, how are you?
All right, good. So I think on behalf of Winship and the Puri family, we'd like to present you with this a plaque to represent the inaugural Puri lecture, and we really appreciate your being here.
Thank you very much, Sagar.
Thank you.
Appreciate
All
it.
right,
Thank you. So kind of you. Thank you.
All right, so there's food outside. Thank you so much.